Welcome to All Force e-learning. Now today we are going to discuss the topic that is gastrulation. In this session we will discuss about the topic gastrulation. Now before going into the gastrulation process, a quick recap what we have studied in the previous class for the formation of bilaminar embryo. Now during copulation or coitus, the sperms are released into the vagina. Now they have moved through the cervix into the uterus and through the fallopian tubes into the ampulla, where the fertilization has taken place. Now this fertilization resulted in zygote. Only one sperm passes through monospermic, cortical reaction takes place, zona pellucida become toughened. Okay. Now sperm license passes through the corona radiata, zona pellucida into the cytoplasm of egg. The fertilization results in zygote. Now zygote undergoes division. This division is unequal. Now holoblastic. Now results in a large and a small cell. This is first division. Then 2 cell to 4, 4 to 8, 8 to 16, 16 cell. 8 to 16 cells is called as morula. Now morula passes through this end of the fallopian tube into the uterine wall. Not uterine wall, actually into the uterus, where it changes into blastocyst. Now this is what happened in this process. Now this blastocyst has the following structure. It has a outer cell mass, inner cell mass. Now outer cell mass is called as a trophoblast or trophectoderm. Now this is inner cell mass or formative cells or embryo proper. This is also called as embryo proper or embryoblast inner cell mass, because it is that part which actually the inner cell mass gives rise to entire embryo. Then zona pellucida still it is present. Then these zona pellucida get digested. This get attached to the uterine wall. Now this part, this is that part. This part is placed here. Now it means the hatching out of this blastocyst from zona pellucida and the outermost layer trophectoderm. Now it get attaches to the uterine endometrium and get embedded inside. Now if you observe, there is inner cell mass. This is inner cell mass. Now this is outer cell mass. Inner cell mass, the part where actually it get hatched out and get attached. Now towards inner cell mass it get attached. The opposite end, this is blastocyst. This is blastocyst, this is inner cell mass. Always implantation takes place towards the inner cell mass. Okay. Now it means this area, this inner cell mass, this area of the blastocyst is called as embryonic pole, whereas this opposite end is called as abembryonic pole, or animal pole or vegetal pole. This part is called as embryonic pole, where inner cell mass get attached, whereas the opposite end, this end is called as abembryonic pole. Now this get embedded. Now it enters inside. Now this is the uterine endometrium. This is basal layer, basal cells of the endometrium. Now normally this is having uterine endometrium, then myometrium, this is perimetrium. Now we are discussing only this part, endometrium. This endometrium is placed here. Now the part where it get hatched out and attached and moves inside. Now this part is what we are saying basalis decidua, uterine wall. After implantation we are calling the name that is decidua. Now this decidua, this is basalis, decidua basalis. Now this is towards lumen, capsularis, this is decidua capsularis. Now this end, the parietal layer, this is decidua parietalis. Now when we observe this one in its development, now the inner cell mass is arranged in the form of, this is upper tall cells and there is a lower cuboidal cells. Now upper tall cells, they are called as Epi Blast. This is embryonic ectoderm. Now the lower cells, this is what we are saying hypoblast. This is embryonic endoderm. These are the two layers in which the inner cell mass is arranged, and such embryo is called as bilaminar embryo. Now, this is bilaminar disc. The inner cell mass, in course of time, the cells are arranged in the form of upper tall cells and lower cuboidal cells. These are columnar, these are cuboidal. Upper tall columnar cells are called Epi Blast. Now, which is that Epi Blast gives rise to the entire embryo, the 3 germ layers also. Whereas, this is Hypoblast, the lower cells. Now, these 2 layers is what we are saying bilaminar embryo. This is bilaminar embryo. Now, next what happens here is these hypoblast, the cells of the hypoblast, they get detached. Now, this detachment of cells of hypoblast and forming a layer surrounding a sac like structure it forms. This is what we are saying yolk sac. Yolk sac. Now, it means by a process called as delamination. Delamination. By a process called as delamination. D means removal, lamina means layer. In the form of a layer, the cells are detached. Which cells are detached? Hypoblast cells. Hypoblast cells, the lower cells, they detach and form a layer. Now, this is called as yolk sac. Now, formation of yolk sac is by delamination, and the yolk sac is formed. Now, towards epiblast, some of the cells which are epiblast cells are called as amnioblast cells. These are some of the epiblast cells on the upper side of the epiblast towards this villi like structure. What we are saying, these are syncytiotrophoblast. Already we have discussed about. This is cytotrophoblast, this is syncytiotrophoblast. With the syncytiotrophoblast, it actually get attached. Syncytio means nothing but having many nuclei, because the cells here are fused. Now, these are the all are cells. Now, all the cells are, their membrane get disappeared, only the nucleus is present. Hence, we are saying this is many nuclei, hence syncytiotrophoblast. Now, towards syncytiotrophoblast, above the epiblast, this is above the epiblast, that is below this syncytiotrophoblast. Now, some of the cells from the epiblast form amnioblast. What the cells are called as amnioblasts. Now, these amnioblast cells, they divide and redivide and form. This is a cavity, amniotic layer with amniotic cavity. Now, it means 2 fetal membranes already formed. This is amnion, this is yolk sac. Yolk sac is formed by delamination, whereas amnioblasts, they form this amniotic layer. Now, still there is epiblast here. Now, such embryo, which is what we are saying bilaminar embryo. This is a bilaminar embryo. This is having 2 layers, epiblast, hypoblast. Though hypoblast formed yolk sac, it is still there. Now, such a bilaminar embryo which is formed, it will further undergo differentiation. Now, that process of differentiation is what we are studying under gastrulation. Now, gastrulation is the process by which the dynamic process it is, means always taking place some changes. Now, hence we are saying dynamic process. Gastrulation is a dynamic process, in sense it is always undergoing some changes. What are these changes? These changes are called as morphogenic movements are taking place. It means the cell or a group of cells are moving to their definite positions for forming a 3-layered embryo. This is what we are calling as a trilaminar embryo. It means formation of 3 germ layers. The formation of 3 germ layers takes place only during gastrulation. What we are saying here, what is gastrulation? Gastrulation is a dynamic process, means undergoing changes which results in movement of cells or cell masses to their definitive positions for the formation of what we call it as 3 germ layers. What are those 3 germ layers? Ectoderm, mesoderm, endoderm. Now these 3 germ layers, trilaminar embryo is formed during gastrulation. If there is no gastrulation, there will be no formation of germ layers like ectoderm, mesoderm, endoderm. Now we have stopped here up to here in the previous class, that is a bilaminar embryo. Now today what we are saying here is these cells, what are these cells? Epi blast cells towards upper side, they form amnioblast cells and forming an amniotic cavity, whereas hypoblast cells they divide and they get detached. This is called as delamination. D means removal, lamina means layer. In the form of a layer, some of the cells are removed and form yolk sac. Now this is called as extra embryonic coelom. This cavity is called as extra embryonic cavity. Now if you observe this bilaminar embryo, it undergoes gastrulation. In this process what happens? Now if you observe this bilaminar embryo, we are weaving, we are cutting here and placing this and we are weaving from the top, that is from the top. This is a ball. Now from this we have removed in the form of a cap. We have made a cut and we are weaving from the top of the bilaminar embryo. What are the changes which are taking place? What are the changes which are taking place? We are weaving from the top by cutting in the form of a cap. Then what we observe here is, this is the layer. What is this layer is? The blue one. This is epi blast, which are columnar tall cells. Now this is hypoblast. These are the 2 layers. Now in the process of gastrulation, the first step is primitive streak formation. Primitive streak. Now primitive streak is formed during gastrulation. What is primitive streak actually? Now this is epiblast, the blue one, what we have shown. Now on the epiblast, there is a small streak appears, now which moves forward. What is this streak is called as primitive streak. Primitive streak. What we are saying here is, when we cut this one bilaminar embryo and observing from the top, what are the changes which are occurring in the bilaminar embryo during this process called as gastrulation. This is what we are weaving. Now when we observe this epi blast cells, the blue colored epi blast cells, these epi blast cells above at the middle there occurs a small primitive streak. Primitive streak is nothing but a group of cells forming in a line. This is a primitive streak and it forms in front. There is a primitive node. This is primitive node. Now this primitive streak which it forms, the cells in the primitive groove, the cells in this primitive streak actually, primitive streak, they die. It means they get degenerated and forms a groove. This is a groove. This is a groove it forms. And at the primitive node, it forms a small opening. This is called as primitive pit. A primitive pit is formed at the primitive node, and this primitive streak forms into primitive groove. This is forming primitive groove. Primitive groove. Once again I repeat here, what we are saying is, first during gastrulation process, a primitive streak appears. A primitive streak appears on the epiblast. This is the epiblast cells. A primitive groove appears. Primitive streak appears first. Nothing but a group of cells in a line is what we are saying, primitive streak. Now this primitive streak in front, there forms a primitive node. a bulging at the anterior. Now it means here differentiation has occurred. Now this area is what we are saying head end, head. Now this is a tail end or anus end or anal end. This is anal end, this is head end. It means this is cephalic plate. Now this is a caudal plate. Now because this part it forming here primitive node, a bulging at the anterior for the formation of head, cephalic plate. Now this end it forming caudal end, that is tail end, it means anal end, this end. Now this primitive streak which was formed, some cells, some of these cells they degenerate, means death occurs, which results in the formation of a primitive groove. This is primitive groove. Now in the primitive node there will be a pit will be formed. This is called as primitive pit. It means primitive streak and primitive node is made into primitive groove and primitive pit. Now what happens? After this formation of primitive groove and primitive pit, now these cells, what are these cells which are present on the epiblast? These are nothing but epiblast cells. These cells are nothing but epiblast cells. Now these epiblast cells, they show growth and they get detached. The epiblast cells, these are the epiblast cells, they grow in number, they get detached and they move from here into the primitive groove. This is the movement shown by the epiblast cells, and they through the primitive groove they move in, they move through this primitive groove and fall on the hypoblast. They fall on the hypoblast. Now this is the hypoblast, they fall on the hypoblast. Listen what I am saying. What are these cells are called as? These cells are called as dash. These cells are called as epiblast cells. The cells which are present on the epiblast, these cells they move and they enter into the primitive groove. They enter into the primitive groove. This is the primitive groove, and then they fall on the, it means here for suppose there is a primitive groove, they fall on the, this is what we are saying hypoblast, and this hypoblast is changed into now endoderm. Endoderm. This is how endoderm is formed. The first germ layer formed is endoderm. How this endoderm has formed? The epiblast cells from this epiblast, the cells from the epiblast, they have moved through the neural groove. They have moved through the neural groove and they have fallen on the, what is the structure? Hypoblast. This is that hypoblast. They fallen on the hypoblast. Now the hypoblast is no more. Now this hypoblast is called as endoderm because the cells were replaced by epiblast cells. Later, some of the epiblast cells after formation of endoderm, some of the epiblast cells again they move through the neural groove and the lateral side and they move in front, forming the middle layer. What is this layer is called as? This layer is what we are saying, the red color layer, what we call it as mesoderm. Listen what I am saying. First what happened here is some of the epiblast cells they have moved through the groove and fallen on the hypoblast. The hypoblast is replaced by endodermal cells. A layer called as endoderm is formed. Then what happened? Some of these again the epiblast cells they move through the neural groove. They move through the neural groove and they move forward and formed another layer. What is this layer is called as mesoderm. Now the remaining layer which is there, this is called as now ectoderm. The remaining epiblast cells together form ectoderm. First endoderm is formed, then mesoderm is formed, finally ectoderm. Ectoderm is nothing but the remaining epiblast is called as ectoderm. This is how 3 layers are formed, that is endoderm, mesoderm, ectoderm. This is what we are saying trilaminar embryo it is now, because of the formation of this primitive streak and primitive groove and primitive pit, it is possible. The formation of trilaminar, this is gastrulation, is the process by which 3 germ layers are formed. This is the 3 germ layers are formed. Simply what we are saying is, after the formation of primitive groove, some of the cells of the epiblast moved through the primitive groove, fallen on the hypoblast. The hypoblast is replaced by endoderm. Then some of the cells of epiblast again moved through the neural groove and moved forward, resulting in the formation of mesoderm, which is present in between endoderm and epiblast. Now, after formation of this mesoderm, the remaining cells of the epiblast are called as ectoderm. This is endoderm, mesoderm, ectoderm. This is trilaminar embryo is formed. Now, this formation of this trilaminar embryo, that these 3 germ layers, is what we call it as gastrulation. The morphogenic movement of the cells through the primitive groove. This is the primitive groove. Provide places or avenues for the formation, because these cells move through this primitive groove. Okay. This is called as ingression. Ingression. What is ingression? Ingression is the process by which the epiblast cells moves, resulting in the formation of endoderm and mesoderm. What is ingression? Ingression means the movement of epiblast cells through primitive groove, through primitive groove, results in the formation of endoderm and mesoderm, results in formation of endoderm and mesoderm. This is called as ingression. These cells, epiblast cells, has ingressed through the primitive groove for the formation of endoderm, mesoderm, and the remaining cells are called as ectoderm. Now, after the formation of these 3 germ layer, the next step is formation of notochord. Notochord formation takes place. Now, here this is the ectoderm. This is ectoderm. Now, it is called as ectoderm. Now, this is the ectoderm. Now, this is the ectoderm. This is the ectoderm. Now, this is that mesoderm. Now, this is the endoderm. Now, we are placed this one like this for our convenience, for understanding what happened here. Some of the, now we are explaining here notochord formation of notochord. Now, how this notochord has formed? Here, look, this is the primitive pit. Now, this is shown here, primitive pit, this is primitive pit. Now, this is the ectoderm, now this is the mesoderm, this is the endoderm. Okay. Some of the chorda mesodermal cells which are present on the ectoderm, some of the chorda mesodermal cells of the ectoderm, because ectoderm is formed, now no more epiblast, ectoderm. The chorda mesodermal cells of ectoderm, they move through this primitive pit into the mesoderm, that is in between, this is endoderm, ectoderm. They move forward, this is, they are moving forward. Simply what we are saying is, this is ectoderm now. Some of the cells of the ectoderm, they converge. Converge means all of them migrate towards one side is called as converging. Now, these cells, they move from all these places. This is from the chorda mesodermal cells move, and they, what we are saying, converge and involute through, they converge and involute through this primitive pit and moves forward, forming, this is what we are saying, notochord. Notochord. Now here is, this one is what we are showing here, this part. If this part is placed here, like it looks like this. Notochord formation. How notochord formation takes place? The carda-mesodermal cells on the ectoderm, they converge, they involute through primitive pit and move forward, resulting in the formation of notochord. Now notochord is present here in this place, mesoderm place, in between this ectoderm, endoderm. Now this end is what we are saying, cranial end, cranial plate. This is caudal plate, this is anal end, that is head end. Okay, it is moving forward. This notochord is the embryonic axial skeleton, is actually the first skeletal structure of the embryo. This is notochord. Notochord is formed like this. The formation of notochord makes another structure to form. What is that structure? That structure is nerve cord. This is called as neurulation. What is neurulation? Neurulation is the process by which nerve cord is formed. Now nerve cord is formed by a process called as induction. Now notochord formation is called dash. Notochord formation is called, yes, the notochord formation is called as involution. What we have said? Involution. What is involution? Involution. What is involution? Involution means some of the carda-mesodermal cells, they converge and move through primitive pit and move forward. This is called as involution. Involute. Now involution means some carda-mesodermal cells converge and involute. Converge and involute through primitive pit and move forward. This is called as involution. Ingression is the process by which endoderm, mesoderm are formed. Involution is the process by which notochord is formed. Notochord is formed by involution, whereas endoderm and mesoderm are formed by ingression. Whereas induction is the process by which nerve cord is formed. This is here the ectoderm. This is the ectoderm consists of cells related to nerve cord formation. It divides and redivides. Now this is notochord is formed. Until notochord is not formed, nerve cord will not be formed. It means notochord attracts the formation of nerve cord. This is called as induction. Induction means the attraction of the notochord to form nerve cord. Now it means in induction process what happens? This is what we are saying, ectodermal layer. This ectodermal layer, it undergoes a small groove is formed. Now these are called as neural folds. These are called as neural folds, and this is neural groove. This is called as neural groove. Now the cells which are present above the notochord, which cells? Ectoderm cells. The ectoderm cells which are present above the notochord. What is this structure? This is notochord. Notochord already formed. This notochord induces. Induces means it calls here. What it calls? It calls the cells of the ectoderm to form the nerve cord. I am already formed. Come here, you also form. This is what the notochord says to the ectoderm. It means here this ectoderm, it undergoes here what we are saying, a groove appears. This groove deepens. And then the neural folds on either side, they get attached with each other, resulting in this is formation of what we are saying, nerve cord. Above the notochord into the mesoderm. It means now notochord is formed as well as nerve cord is formed. The formation of nerve cord is what we are saying induction. What we are saying induction. 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 Induction means the process by which the notochord attracts some of the cells above the ectoderm to form a neural groove. Now this neural groove deepens and these neural folds they get attached to form a nerve cord. Now it means it looks like this. Here this is notochord. Now here there is nerve cord. These two structures appear here in the embryo. Now hence the notochord is formed. Now the nerve cord is formed. Now it shows here bulging at the anterior side. This is what we are saying for the formation of head. The nerve cord shows bulging towards the anterior end because that is the cranial plate where the brain formation takes place. Now this is the caudal end, that is tail end or anal end. That is anterior, this is posterior end of the embryo. This is how three germ layers along with notochord and nerve cord are formed. Okay, this is how this is formed. Ingression, involution, induction. Ingression, movement of epiblast cell through primitive grooves to form endoderm and mesoderm. Involution, some of the carda mesodermal cells converge and then involute through the primitive pit, moves forward to form notochord. Induction, some of the epiblast cells are attracted by notochord, forming a primitive, this is a groove, neural groove. Neural folds get attached, a nerve cord is formed just above the notochord. The first structure, axial skeleton, the skeletal structure which is formed is notochord. Now upper one, this is nerve cord. Now this is mesoderm, this is ectoderm, this is endoderm. Okay. This is the process of gastrulation, notochord formation, and then there is nerve cord. Okay. Okay, thank you. We will discuss in the next class the next topic.